can use that to recreate a complete copy of the double helix. So these two strands, they have to pull apart, but this is actually quite a complicated and difficult procedure. And the reason is that DNA is incredibly convoluted within our cells. So each cell nucleus is typically about 5 or 6 microns across. And if you were to take the DNA that's just sitting in that cell nucleus and stretch it out into one long line, it would be 2 meters long. So something 2 meters long has to end up by getting scrunched up into something just 5 microns across. So it's incredibly knotted. And when the cell replicates, these 2 bits have to somehow get from one has to end up in one cell and the other one has to end up in the other. So clearly, the 2 strings have to pass through the cells, pass through each other. And this is done by various enzymes, and importantly, the enzymes that work for bacterial DNA are different from the enzymes that work for human DNA. So in fact, lots of the antibiotics that we use, they specifically block this untangling procedure in bacterial DNA. And so that means that the bacterial DNA cannot unknot itself, and so cannot replicate, and so dies. Whereas that has no effect on us because we use a different type of enzyme to do this. So clearly, understanding this unknotting procedure is important, but it's not completely well understood. And there's been a very productive collaboration between biologists and knot theorists to be able to understand that in more detail. So that's just one example of the occurrence of knots and the importance of knots in nature. I'm a pure mathematician, and one of the reasons that I find knots interesting is because they are